like we do most times, on the non-specific defenses. So one of the take-home organizational tools I want you to use is this breakdown of the immune system. Okay, so the immune system is made up of the innate, non-specific, and the acquired specific. So last time is when we really hit these, the non-specific defenses, right? So we talked about the skin, and in addition to the skin as a physical barrier, you also have things like the mucous membranes, the secretions, so saliva, sweat, mucus, all are physical barriers, okay? Even though they're produced by living cells, they are themselves not alive, okay? So these physical barriers are those systems that are, physically a barrier to infection getting in. We talked about the phagocytes. There are mobile ones and there are stationary ones. The mobile ones are the white blood cells. So they travel around the body looking for infection, debris, pathogens that don't belong. And then there are the stationary ones that we call fixed macrophages. So they live in the tissues all the time. And they just sit there doing nothing unless there's an injury or unless there's infection. So the skin and the mucous membranes have a bunch of these fixed macrophages because those are places where infection or trauma is common, right? So they're there to do their job. We talked about immunologic surveillance, the natural killer cells, how they're on the lookout for mutant cells or virus-infected cells. Those are the two populations that they look for most, okay? And they're a form of T cell, the natural killer cell is. Interferons protect other cells from virus infection. The complement system pokes holes in bacteria once it's activated. And then we ended last time talking about inflammation. So that redness, swelling, warmth, and pain, you should know those 4 words for sure, right? Redness, swelling, warmth, and pain. And then a little bit about what causes those. So histamine in particular causes vasodilation, and which causes the redness, the swelling, and the warmth, and it also is part of that pain system. So the only one we didn't get to last time was fever, okay? And fever is oftentimes misunderstood. Fever is not a bacteria's fault or a virus's fault, okay? Fever is something your body is doing on purpose in order to fight off infection. So sometimes people have a tendency to think that the pathogen causes the fever and that somehow that's bad. It's not that. It's the response of our body to infection is to elevate the temperature. And you might think, well, why on earth would we do that? Well, when you have a fever, number one, you knock the pathogen out of its ideal range, These bacteria in particular, as many of you will have learned in micro, have this very narrow temperature tolerance. If it's too hot, they don't grow very well. If it's too cold, they don't grow very well. Well, if we could somehow drop our body temperature, that would be even better of a defense than fever. But as warm-blooded creatures, we can't really do that because we're creating heat all the time. So we can't chill the bacteria and slow their reproduction, but we can heat them up and slow their reproduction that way. So fever is not just a symptom of infection. It's also the body's attempt to treat that infection or that cause. So what you all will find that when you take care of patients, whether they be human patients or non-human patients, is that there's this tendency we want to stop the fever because we see the fever as a symptom or a problem. Fever is inconvenient because it makes people really sleepy. It is metabolically expensive, because you burn through calories faster, you burn through fluid faster. But the truth of the matter is, other than its inconvenience, fever is actually a benefit because it slows bacterial growth down. It helps the immune system to function better at that higher metabolic rate. So even though we have a tendency to want to treat fever, the truth is fever is a natural occurrence as the body fights off infection. So what does that mean? It means don't treat somebody with a fever just because they have a fever. Okay? If they have, if they absolutely have to get to work, or they are not drinking well because they're so sleepy, or it's gone on for a long time, okay, fine. Treat the fever when during the day you need to. But typically speaking, fever is healthy. So don't treat it. Let it run its course, and the infection time is usually less. All right? So, here's a little girl with fever, and something about that look makes us want to fix that, right? So we give them the medicine, the fever comes down, they look better, but we've knocked out one whole non-specific defense for them. So now there are some exceptions. Fever in kids have higher fevers than adults do. So if you see 104 temperature in a kid, don't freak out, that's fine. The smaller the creature, the higher its temperature is usually anyway. Anything above 105 in an adult, okay, now you're at risk for seizures, brain-related high temperature stuff. In kids, we usually push that range up to 107 or 108. The other exception is if you have a person who has seizures when they have fever, okay, then you treat the fever to prevent the seizure. So just a little bit about fever. And that technically a fever is just a rise in body temperature above normal, and it is the body's fault. Now, it's not the bacteria or the infection that causes it. The body raises the thermostat. So just like in your home, you can go over on a cold day and you can ratchet it up to 80 degrees, and the heater will pump and make it 80 degrees. That's what's happening with the fever, is the body has raised its set temperature. It's why you can't stop a fever with cold. People used to think this, that, the child has 104-degree fever, put him in the bathtub, fill it with ice. Worst possible thing to do, because the body, that kid, is going to keep trying to keep their temperature at 102, and you're dumping ice on them. So essentially, you're giving them this huge metabolic challenge, and shock can often occur. So cannot treat a fever with cold. It's the body setting its temperature. About the only thing you can treat it with is medication. So Tylenol will certainly treat a fever, aspirin, and then the whole class of medicines called NSAIDs, non-steroidal anti-inflammatories. That's ibuprofen, naproxen, and all of its friends and family. All are good medicines to treat fever. Did I see a hand up? No. Okay. So a little bit from the pediatrician on fever and how it's sometimes just fine not to treat it. Yes?
So the dangerous level is just because the body will overheat and just kill too
It's the
much.
brain. The brain is most sensitive to high temperatures. And when temperatures get too high, it can have little seizures, which are not terrible, but a seizure that goes on for 15 minutes or more can cause brain damage. Plus, seizures, you think fever freaks people out. Seizures will level the room. Everybody will be freaked out about that. Yeah.
So what about for babies? Are their temperatures going to be even higher?
They can, kids and infants can make a higher temperature faster. So like, for an adult, you'd have to be really darn sick to get to 105, 106. Babies can do that in an hour because they're so much smaller. Their fever system works better. So they can make a better fever faster. Does that answer your question?
Oh,
yeah. And, in kids, one of the things you worry about with fever is dehydration. Because when kids have fevers, you all have probably seen this, they get very lethargic, right? They just want to lay and do nothing. Well, a lethargic child is not drinking, and a feverish child is burning through water faster. So one of the reasons we will treat a fever in little kids is to avoid dehydration, but that's only if it goes on day after day. Yeah.
Sorry, one more question.
No, light questions.
Why is it so, why is like when the quote-unquote fever breaks, why is that so uncomfortable? Like you just sweat a lot, and like you can tell when your fever is quote-unquote breaking.
It's because the body's been trying to make you hotter and hotter and hotter. And then at some point, the switch is thrown, And now it wants to be back to its normal temperature again. So you have all this heat to get rid of. So all of your heat losing strategies kick in. So you sweat, you don't want to have anything on you, you want to be in a cool room. It's all to get rid of that extra heat that you developed.
And that's kind of a sign of the infection being killed?
Yeah, usually it's either the infection is on its way out, or at least the load has gone down enough where the body's turned the fever signal off.
Okay.
Now it can come back on. In a lot of diseases you get this, where the fever breaks and you come back to normal and then it builds back up as the infection enters the bloodstream and then is cleared from it. It enters it again and is cleared from it. Yeah, good questions, yes.
So why do you feel really cold sometimes when you have a fever?
Because your thermostat's been turned up. So you're sitting there at 98.6 degrees, but your brain says, I want to be at 101, so you feel cold. So you do all the things that you do when you're cold to build that heat back up. So you do this, you shiver, because shivering produces heat. You want to be covered up. So it's the opposite of the fever breaking thing. Yeah.
I have two questions. What's the mechanism for your brain to do that, to kind of switch it? Because that seems pretty dramatic.
It's the hypothalamus responding to a whole class of inflammatory mediators. There's prostacyclins in there, there's prostaglandins in there, some of those eicosanoids that we talked about in the endocrine system. So when the hypothalamus senses those inflammatory mediators, it kicks the temperature up. And it's in charge of the body's temperature all the time. So all it's doing is changing its desired temperature. Does that kind of make sense?
Yeah, and then it sends out like a response through the blood?
Right, the hypothalamus sends out through the sympathetic and parasympathetic nervous systems the signals to either create extra heat and hold on to heat like you do when you're cold, like outside today, or the opposite, get rid of heat. So heat, and we're going to talk about heat gaining and heat losing in another chapter. But fever just changes the body's desired temperature. So then the rest of the body responds to that.
And I've had patients who they have all the symptoms of fever, but they don't actually have a fever. Are there people who just don't get fevers?
There are people that don't get fevers very well. And there are people whose fever response, there are even people who are flipped.
Yeah.
Where in response to an infection or some other big stressor, their body temperature will drop instead. You see that in infants sometimes. Yeah, like to feel. Yeah, so she just may not be able to generate a temperature. Yeah, it's not a perfect science. It's like, we all read in the textbook 98.6 degrees. Well, that's not everybody's normal temperature. Some people are at 99, some people are at 97. Tends to be the smaller you are, the higher your temperature. But there's a range. That's why you got to compare a patient to themselves, not to the book usually. All right, so that's fever. Some questions to start off today. So each of the following is a physical barrier, except one of those. And we'll put it back. All right, so a physical barrier is something that stands in between a potential pathogen and the tissues of the body. Okay, so that's going to be body hair. That's one of the reasons that we have hair. I mean, we humans have lost most of our hair, but mammals have hair because it's like a second skin. It prevents damage to the skin and therefore infection. Epithelium is always part of physical barriers, as are secretions. And basement membranes. Did you guys talk about basement membranes when you talked about epithelia last semester? That was before I came in. Essentially, all epithelial cells sit on a sheet, a protein sheet, and it helps keep them stuck together. It also acts as a very, it's not very permeable. So the basement membrane, things can't pass through it very easily because the holes are super, super small. So it acts like a physical barrier. So the one that it isn't is complement. Complement is a biochemical protein attack system. So it's not physical at all. It's molecular chemistry. It's biochemistry that makes it work. Okay, the first line of cellular defense against pathogens. So now we're through the physical barrier. Cellular defense is which of those? Okay, so you've got a bacteria and it wants to infect the blood, let's say. So after it's gotten through the physical defenses somehow, usually because there's a cut or something, what is it going to encounter next? And it's going to be the phagocytes, particularly the fixed macrophages that sit underneath the skin and are on the lookout all the time for anything that doesn't belong. Even in a tissue that doesn't have those, say in the in the spleen, for example, if a bacteria gets there, the phagocytes are the first thing it's going to have to deal with. And I added to my list over here the innate, the non-specific defenses which we've been talking about. They're fast because usually they're already present. And if they're not already there, they can be made in a hurry because they're all the same. So here's what I mean, like neutrophils. If you are experiencing a new bacterial infection, your bone marrow can churn out a ton of neutrophils real fast because they're all just carbon copies of each other, okay? The acquired system, they're not all carbon copies. Each one is different. Each T cell and B cell is different, so you can't just churn out a million of them quickly. So these defenses we've been talking about are fast, and they're the first to start attacking an organism. The acquired system is slow, complicated, but in the end it's way, way better. So, for example, you've all had a cold and somebody has said, 7 to 10 days or 5 to 7 days and you'll be better. Well, that time is the time it takes for the acquired immune system to work up a response to that infection. So it's slow. 7 days is slow, but not long after that 7 days and you're feeling quite a bit better. So it's much more effective. This is fast, but kind of limited in what it's able to do, particularly against viruses. Okay, last one. The cells that perform immunological surveillance are the which of those? All right, good. That is A, the NK cells. Immunological surveillance is the body's immune system watching, surveillance is watching all of the tissues and cells of the body. So the natural killer cells are always on patrol. looking for cells that are infected by a virus or have become mutated somehow. And, unlike our spectacular police that we have in America, the NK cells, their only response to a problem cell is they just kill it. So it's very quick and not very pretty, but it does the job, which is to keep virus and viral infection and mutation from spreading. Okay, so that's this branch, the non-specific defenses. And so as we move then into the other branch, we'll see that instead of a long list of different kinds of defenses, the acquired system uses a few key mechanisms to create sort of a custom-made immunity to each and every pathogen that we experience. Okay. So as we break out the specific defenses a little further, we see that we essentially have, two categories. So we have some innate specific immunity, which we don't talk about very much. But essentially, just like we're born with neutrophils that attack bacteria, we are born with some lymphocytes that can create a general attack. Now, we don't talk about it very much because it doesn't come up very much. It's this branch that comes up. So first we have the acquired. That just means you're not born with it. And the easiest way to describe this is the acquired side of the immune system has to learn about a pathogen before it can do anything about that pathogen. Okay, and we'll talk about some of the mechanisms through which that happens as we go. So inside this immunity that we learn, we have two big categories. So we have passive immunity and we have active immunity. Okay, so what occurs in the natural case is this right here, naturally acquired active immunity. This means somebody coughed on you, you got a cold, and then 7 to 10 days later, you were cured of that cold. You wiped out that virus. The doctors were not involved. There was no vaccines involved. There was no medication involved. That's your immune system did it all by itself. So this occurs, all the time. We are always fighting off pathogens. Each and every day, something is trying to infect you. Now, the reason that we're not sick all the time is because we have an immune system that takes very good care of us in our very diverse and dense planet Earth where there's all kinds of tiny little living things. Okay, so that's the natural. Well, what about immunization? In immunization, we trick or teach the immune system about a pathogen without actually exposing the patient to that pathogen. Okay? So we call that, the fancy name is artificially acquired active immunity. Okay? It's active because your body made it. Okay? It wasn't given to you from somebody else. But it's artificial because we've whipped up this concoction to inject into you. So instead of being exposed to the virus, we gave you parts of that virus is usually how it's done. So essentially, immunization works. In that vaccine are molecular pieces of a virus or a bacteria that allow the immune system to identify that virus or bacteria if it ever sees that in the future. Okay, so like take measles, for example, because that's been in the news. So in that vaccine are pieces of the measles virus, not the measles virus, or at least not the active measles virus. So the immune system learns about those pieces, and then when it sees the real measles virus, it knows that that's a bad thing and attack it. Okay, so it's like pre-teaching the immune system about a pathogen before exposure. All right, so we'll talk more about vaccines as we go. But that's artificially acquired active immunity. Now these over here, the passive ones, these don't come up very often. Essentially there's two forms. Passive immunity just means it's immunity that you didn't make yourself, but ended up in you somehow. So examples are key here. The naturally acquired version of this is in breast milk, mom's antibodies, so an immunity that mom made, is transferred to the baby. through the digestive tract. That means that as long as baby is breastfeeding, baby is sharing mom's immune system. And antibodies are the silver bullet of the immune system. Once you have antibodies against a pathogen, you're going to be immune to that pathogen. Because antibodies, as we'll see at the end of our talk next week, or whenever, antibodies make the whole immune system work as one to destroy anything that has that antibody attached. So that's a version of naturally acquired passive immunity. This also happens in fetal development. Antibodies cross the placenta. So mom's immune system is looking out for baby while baby is in uterus. And then the rarest of all of these things is this one, artificially acquired passive immunity. This is where we inject you with antibodies made some other way, either made in another person, which they used to do a lot but don't do much anymore, or made in a laboratory. Now, why on earth would you need that? The most common example is sort of a bizarre one, but it's snake venom, all right? So you're traveling in Arizona and you get bit by a diamondback, right? The way that they keep you from dying is they give you antibodies to diamondback venom, and then the immune system attacks that venom and destroys it so that you don't die. So that's an example of artificially acquired passive immunity. I told you it was sort of rare. Another place we see this sometimes is in patients whose immune systems don't work right. They sometimes will get antibody infusions to help keep them healthy. Again, these things, they used to be done a lot, but then with all the blood-borne pathogens that there are today, to get antibodies from people, you're usually not getting just one person's antibodies. You're getting like the pooled antibodies of 1,000 people. So it's like getting 1,000 blood transfusions in one in terms of risk for blood-borne disease. That's why we don't do this stuff very much anymore. Plus, now we can create antibodies in the laboratory. But the most important of all of these is this guy right here, because this is the natural case, which is what we're going to talk about the most. I will talk a little bit about vaccination just because it's a hugely important public health issue, as we've all heard about in the news, so we're going to talk about it too. Okay, so before we can get into the acquired immune system, we need some properties. You know, what does it have to be, or what characteristics does it have to have in order to do what we need it to do? Because ideally, we want an immune system that will protect us from any and all pathogens, right, as quickly as possible, all without ever attacking a normal, healthy cell, right? So we're asking a lot from a system that has no brain. You know, the immune system the brain has nothing to do with it. It is a bunch of cells doing different things that create this system. Okay, so first we want specificity. What this means is not all T cells and B cells are the same. In fact, they're all different on purpose. Okay, so specificity says we want one specific pathogen to activate one family or specific line of T cells or B cells. Okay, so much more on this later, and we're going to talk about what an antigen is. So we'll get back to specificity. Versatility, we want to be able to defeat any and all possible pathogen. So the system has to be versatile. You know, bacteria A is different than bacteria B. Virus A is different than bacteria A and B, and viruses B through Z, right? So our immune system has to be able to handle each of these different pathogens that's present in our environment. Okay, so we want versatility. We want memory. Ideally, we would only get infected by a thing once, right? Because what's the point of getting the same infection over and over again? So we want our immune system to remember and learn so that if we got chickenpox when we were little, that we don't get chickenpox every time we're exposed to it. Okay, so we want immunologic memory. And then finally, we want tolerance. The immune system is, first and foremost, a killing system. You know, it kills cells. It kills bacteria. It kills viruses when it can. Well, if we're going to have normal, healthy tissues, our immune system must never attack a normal healthy cell and destroy it, right? Otherwise, our own immune system has turned against us. We call that autoimmune disease, and it does happen. But in the perfect case, we want our immune system to always tolerate self. We're going to call it self and non-self. Because this system is made up of cells, T cells and B cells. T cells and B cells are not very smart. So we have to think in very simple terms to understand what they're doing. And one of the things that the acquired system does is to discriminate self, in other words, normal healthy tissue cells, from non-self, anything else, right? So that might be a virally infected cell, might be a bacteria, might be pollen, might be somebody else's cell that ended up in your body somehow. So we need to have that tolerance of self and versus non-self. Okay. So, in general, and we're going to get to the details, is, this is the in general immune response, is there are, pathogens appear in the, let's talk about antigens here real quick, because that word is all over this slide. Did you talk about antigens last semester? Probably. All right. So my people in here that have had immunology, I apologize for the silly simplicity for which I'm about to do this, but it works, okay? All right, so here is our friend bacteria, right? Now, when we've looked at these under the microscope and we see these little tiny shapes, well, if we zoom way in to, the side of the bacteria, it's anything but simple. You've got proteins hanging out here, all different shapes and sizes. You've got circles and squares and all those kinds of things. So remember that a bacteria is essentially made up of molecular pieces, right? Well, this is a big thing. To our immune system, a bacteria isn't so small. It's a large, too big to identify all at once. So the trick that our immune system uses is it turns pathogens into antigens. So here's our intact bacteria. Here, I'm going to put some shapes in here, okay? So we got a star, a square, a triangle, a circle. If we take this bacteria and we smash it into little pieces, right? Okay, so we take it, we put it on the animal, and we go whack. Well, what are we going to have scattered around? We're going to have pieces of bacteria scattered around, right? So the square, the triangle, the star, and the circle, which used to be part of the bacteria, have now been separated from their original spot, right? So like here's a triangle, here's a star, here's a circle on the membrane. So these are antigens. So an antigen is just a molecular piece of a thing. Now, it can be a piece of anything. Our cells have antigens. So like, if you took a red cell and you smashed it with a hammer, you'd have little bits of pieces of red cell, right? And some of those pieces would be unique to you as an individual. Some of those pieces you'd find in every human being on the planet. Okay, so antigens, that word just means a molecular piece of a thing. And we use, the immune system uses antigens to identify pathogens, all right? So, immune system's on the lookout, and it finds a square. Well, by previous exposure, the immune system knows that that square is part of this bacteria, and that it will respond anytime it sees this bacteria because the bacteria has a square in it, and it knows about the square, right? So that's kind of the antigen system. So what we see here in our overview is these pathogens end up being processed, in other words, smashed into little pieces, by the phagocytes. The phagocytes pull those antigens out, teach the immune system about those antigens, and then we get an antibody response and we get a cell-mediated response. And in the end, that results in destruction of the antigen, as well as anything that antigen is attached to, or that that antigen is a part of. So like in my example, once the immune system learns how to destroy square, it will destroy bacteria because bacteria has square attached, has square as part of it, right? Okay. I know it's sort of a, oh, here's my slide. So I like to use this analogy right here, and maybe it's silly, but it works for me. So these cells can't see or hear, but in some ways what we need them to do requires them to have some senses. How is it going to tell that this is a bacteria and not a normal epithelial cell? Well, it has to have some way of sensing that. So the antigen system is kind of like vision for these lymphocytes that make up the acquired immune system. It allows them to feel a piece, because it's really more of a touch system, but they feel a piece of a bacteria and then know that that bacteria is something to be destroyed and not, for example, a normal epithelial cell. Okay, so this is all just saying what I said there, but with letters and stuff. So good, another way to look at it. And then the hearing part, these cells have to be able to get where we need them, okay? And they obviously don't have ears, and our cells don't make sounds. So the hearing system is these cells can follow a chemical message. So they can get where they need to go. Like we talked about chemotaxis with the white blood cells. These cells use that chemotaxic sense to get where they're supposed to go, to get where there's damage or infection happening. All right. So, of course, it's not as simple as that, right? Once we have antigens, we have to do something with them in order to teach the immune system that square is something that should be destroyed because it's part of bacteria, okay? And that process is called antigen presentation, which is where an immune system cell takes one of those antigens, packages it in a signaling molecule, and displays it to the immune system so that the immune system can then learn from that piece, all right? So because, as I say here, not all antigens exist outside of cells. Some viruses, for example, live their whole lives inside of cells. So the body needs to have a way of identifying, first, is this antigen something that should be destroyed? And second, is that thing to be destroyed inside of you cell, of a particular cell? So to make this system function and not overfunction, in other words, not destroy normal healthy cells, we have this system called the major histocompatibility complexes, the MHC, big long word. What is this histocompatibility? This is, you all have heard in transplants how the organ has to be matched to the donor, right? That is the compatibility that this is talking about. Because we're all individuals, we all have unique genes. That means that many of our cell parts are not identical to, our neighbor, for example. But the MHC molecule is particularly diverse. In other words, the only two people that have the same MHC molecules are identical twins. Every other person on the planet has a different MHC molecule than everybody else. And you might say, well, how on earth is that even possible? Well, evolution has built this in. This molecule is particularly variable. So the genes that make it are particularly variable. So you don't get it from your parents, you don't get it from, you don't get it from some pathogen. I mean, there's inheritance. But in other words, each person has their own MHC. And there's two classes. There's class 1s and there's class 2s. So this is an important point down here. Class 1 MHCs are found on all cells, right? Class 2 MHCs are only found on immune system cells. All right, I promise it will start to fit together, because right now it's a lot of pieces. We've got a lot of antigens right now in this talk, but they're coming together. Okay, so the easiest way is to look at one example. So Class 1 MHC, these are found on all body cells. Well, all cells that have a nucleus, which means not red blood cells, but all other cells. And this not only is going to show you how these packaging molecules work. So essentially, an MHC molecule is like an individualized mailbox, okay? Because we, as individuals, we are all different, so you can think of it as our MHCs all have different addresses. But the message in the mailbox can be the same across different individuals. Okay, so we'll start at the exciting event here. So here is virus coming along, and it has injected its DNA or RNA into the cell. Now, some of that DNA or RNA is, of course, going to go on to become virus in the end. But our cells have a small bit of immune system too. So when they recognize something that doesn't belong, so like here's a piece of a virus, here's a bacteria that's been swallowed, well, the cell destroys those invading things. And in the process of that destruction, we get these little bits and pieces of those things that we call antigens. Okay, so this is also called antigens right here. Those antigens end up packaged in one of these MHC molecules. So here, the message, the antigen, is being packaged in the mailbox here. And then the message and the mailbox both are transported to the surface where they're put out there for display. Now, what comes next, which we'll end up talking about next time, is now the T cells and B cells come along and they look at this message and decide if this is a problem. In other words, if what is in the mailbox, in other words, the antigen that's present in the MHC molecule, if it is one that the immune system has learned is a pathogen, this cell will be destroyed, right? Because now it's got this marker on its surface. It's saying, I've got this inside of me. Along comes the immune system and says, well, that's a pathogen, so off you go, and destroys that infected cell. Okay, so this is the class 1 system. This is happening on in all of your body cells when there is a pathogen. Now, MHC molecules are not always full. This is a case of an infected cell. In a normal, uninfected cell, the MHC is still on the surface here, but this pink pill-looking thing is absent. So the mailbox is empty. So the immune system comes along, checks it, and says, okay, you don't have an infection, and moves on to the next cell. So that a normal, uninfected cell isn't destroyed. All right, so that's class 1. The class 2 is a little bit different because this is the immune system doing this routine on purpose, which is educating the immune system about some pathogen. So now we have, here's our macrophage, right, sitting there in the tissues, and you've stepped on a nail, right? So in comes your tetanus bacteria, right, and it's going to get swallowed up by this macrophage, destroyed into little bits and pieces. So here we have bacteria, bits and pieces of a bacteria. Those are antigens, right? Bits and pieces of a thing. Well, some of those antigens end up packaged in a slightly different mailbox and displayed on the surface of the macrophage. Now, because it's a different molecule, it's a different message. Okay, the class 1 MHC message is, this is inside of me, decide if I should be killed or not. Okay? The class 2 message is, I found this in the body, and I think it's a pathogen. So this is an immune system cell telling the acquired system, all right, ramp up an attack against anything that has this piece as part of it. Okay, so this is a teaching moment. The macrophage is going to teach the T cells and B cells that whatever this used to be a part of is a pathogen and should be destroyed on sight. Okay, so like here they picked a round circle or a red circle. You see that this bacteria has red circles on its outer membrane. Well, once the immune system learns that that red circle is a problem, anytime it sees red circles, it's going to attack that thing. So like this bacteria won't stand a chance in about 5 days because the immune system is going to attack these pieces along the outside. All right, 5 minutes. So this is a little bit of the checking, Okay, so here comes a T cell, and it has its little receptor, its detector, and it's going to look at this antigen, confirm that it's in the right mailbox, and then check this antigen recognition protein, checks to be sure that this whole system is working properly. And let's say it's a match. Then this inactive T cell becomes active and goes on, makes a bunch of copies of itself, and creates an army to fight whatever this antigen is. Okay, so a little bit about T cells and B cells. They each have a receptor that is unique to them. So we have hundreds of millions of T cells and B cells. Each of them has a slightly different receptor than all the others. That means that each T cell and each B cell is essentially looking for one and only one antigen. Okay, and when it finds that antigen, then it's going to be activated and is going to start that acquired immune response, which I have, given a little foreshadowing on. Okay, so, and this is just kind of walking you through that. It has to find, the T cell has to find the antigen that matches its receptor in the right kind of MHC molecule to trigger that immune response. Now, because of HIV and the fact that many of you will take care of patients with HIV, or at least you're going to be asked questions about it on various board exams and such, I have to add a little bit of minutia, which is these last 2 points. Okay, so we have class 1 MHCs and class 2. Helper T cells are T cells that assist the other parts of the immune system with their function. For today, I just want you to make an association. Okay, so class 1 MHCs go with CD8 T helper cells. And class 2 MHCs go with C4 T helper cells. Okay. The way I remember this is they both multiply out to 8, right? So 1 times 8 is 8, 2 times 4 is 8. So the 2's go with the 4's, the 1 goes with the 8's. So what do these names mean? CD4 and CD8 are just names for molecules on the surface of these 2 populations of T helper cells. Okay, and the whole reason that this level of detail has become expected is HIV affects the CD4 cells. So not just those, but preferentially. So like one of the measures of a person with HIV infection is their CD4 count, okay, which is essentially their the T helper cell count of, let's see, did I say that right? I didn't. Okay, these are not helper cells, these are cytotoxic. Correction, these are, no, those are helpers. These are cytotoxic. I'm not an immune system expert, hopefully that won't come through again. All right. So CD8, that's the cytotoxic T cells, much more on that next time. And class 2s go with the CD4s. Okay, so we haven't really put a lot of pieces together yet. I promise we will tomorrow. Okay, we'll start to see how antigens, this recognition system, variability of receptors all come together to make this acquired immune system thing work. All right? This is definitely a chapter where reading ahead a bit is a good idea because there's a lot of stuff in here. It's like a whole course that we're doing in a week. So, but we're out of time. So